So hello, everyone, and welcome today to Grand Rounds. We're honored today to have Dr. Rasheed Gbadegesin present the 11th Annual Dimitri Ellis Lecture. As a reminder for those who are attending virtually, questions can be posted in the Q&A throughout the lecture, and you can join us for an informal meet and greet at the end of the presentation by asking us to unmute you. The CME and maintenance of certification codes are posted in the chat and will be displayed at the back of Rangos Auditorium. So I wanted to just take this opportunity to say a few words about Dr. Ellis. So Dr. Ellis immigrated from Greece to the United States at the age of 13 and really went on to realize the American dream in becoming a pediatrician. He completed his residency here at Children's Hospital of Pittsburgh and returned to Pittsburgh after his fellowship, really to start the Division of Pediatric Nephrology. So he's our inaugural pediatric nephrologist here in Pittsburgh. He taught residents and students and looked after all the children with diverse kidney disorders throughout our entire region, in the midst of also carrying out clinical and laboratory research. So truly sort of a triple threat throughout his time here. Dr. Ellis's clinical expertise is well recognized, particularly in the field of pediatric kidney transplantation. And his patients were the very first children to receive tacrolimus or cyclosporin immunosuppression for kidney transplant here in Pittsburgh. Superior outcomes had distinguished children's as amongst the top pediatric kidney transplant programs, and that continues to be the case. Dr. Ellis was awarded the 2011 Howard Mermelstein MD Award for Excellence in Pediatrics as a recognition of his clinical excellence. Despite his busy clinical life, he also engaged in really high quality scholarly research with more than 100 peer-reviewed publications and 28 book chapters. He served here as the center principal investigator for the North American Pediatric Renal Transplant Cooperative Study while he was here, and was a co-investigator for the epidemiology of diabetes complications, a longitudinal NIH-sponsored study. He officially retired from his position at Children's in 2017, having given 40 years of service to the children in our region with kidney disease. And in celebration of him, we honor him with this lecture series annually. And today we're pleased to welcome Rasheed, who is the Wilbert C. Davis Distinguished Professor of Pediatrics and Professor of Medicine at Duke University. He's also the Associate Dean for Physician Scientist Development and Director of the Office of Physician Scientist Development. His work is really focused around understanding the molecular pathogenesis of nephrotic syndrome, the biologic basis for disparity in its incidence and therapeutic responses. In the last 15 years, he and his team have identified at least 10 new genetic causes and genetic risk factors for nephrotic syndrome and other chronic kidney diseases. He and his collaborators have continued to characterize the mechanisms by which these genes cause nephrotic syndrome and have identified biomarkers of disease and druggable pathways that may treat these defects, as well as the more common idiopathic nephrotic syndrome. He's truly a leader in our national and international networks that are unraveling the genetic basis of excess chronic kidney diseases in individuals of African ancestry. Amongst many other awards, he recently received the Doris Duke Foundation Paragon Award for Excellence in Research. He's published over 150 articles and currently holds multiple and leads multiple NIH grants. And I should say he was excited to order Dr. Ellis's book, which is portrayed here when he was looking in preparation for this lecture. So please join me in welcoming Rasheed for this lecture this morning.
Thank you very much, Jackie. And I tried my possible best to be there in person because, you know, I have so many friends and colleagues there. And in particular, I wanted to meet and, you know, have lunch with the Nephron man. So I feel highly honored to be the speaker for this lecture. And as Jackie has said, Dr. Ellis founded this division in 1977. He retired in 2017, but he's definitely not tired. Dr. Ellis is an accomplished clinician, a role model, a mentor, and a sponsor. We're right to describe him as Professor of Professors, going by the stature of the Children's Hospital of Pittsburgh Division of Nephrology, and the numerous trainees that have graduated from the program under his watch. I have no doubt that my good friend, Dr. Jackie Ho, is carrying on the tradition of excellence associated with this program. And also, all the lecturers before me are all giants in the field, and they are all nice colleagues and friends. What a big honor for Dr. Ellis. So the title of my talk today is Genomics of Nephrotic Syndrome, Closing the Translation Gap. And what I'll try to do today is actually to give an overview of what the genomic revolution has done in improving our understanding of nephrotic syndrome and other glomerular diseases. Then I will talk about recent impactful findings that is actually going to lead to new therapeutics in nephrotic syndrome, and talk about the basically the constraints and how I feel we can actually overcome the constraints. Okay, can I advance my slide? Okay, so most important thing, I have no conflict of interest associated with this presentation. Okay, so I did a very quick search online looking for Dr. Ellis's picture. And the first thing that came out was that when I put his name, everybody that has ever passed through the Division of Nephrology at the University of Pittsburgh were actually labeled Dr. Ellis. So that's very interesting. And then I came across this book. I said, oh my God, Reflections of Nephron Man. Of course, I ordered the book right away. And thank you so much, Dr. Ellis. At least I now have a present that I can be giving to our incoming and graduating fellows. So I guess I can say that we are all Dr. Ellis. Thank you so much, Dr. Ellis, for your contribution to our field. So what I will do in the next 40 minutes is actually to give an overview of clinical significance and areas of need in childhood nephrotic syndrome, discuss recent genetic findings and its impact on understanding of disease mechanism, illustrating this with our recent work. And then actually discuss a collaboration that multiple investigators are, collaborations among multiple investigators to understand one very important genetic predisposing factor to FSGS and nephrotic syndrome, and how focusing on that and trying to understand disease mechanism has actually led to identification of a therapeutic agent that is in clinical trial within 10 to 12 years of the discovery of the gene. And then I will discuss problems and prospects, and then round up. Okay, so let's start with the clinical significance. So when people ask me, what is your research focus? I tell them I do molecular genetics of nephrotic syndrome. And what I usually get is that, okay, fishing expedition. So I came up with this slide. So the way in which I look at it is that we can divide nephrotic syndrome into two broad classes. Steroid-sensitive nephrotic syndrome. This is the most common, seen in 80% of children. And then steroid-resistant nephrotic syndrome, seen in about 20%. And the major problem with steroid-sensitive nephrotic syndrome is that we do treat children with corticosteroid. Majority of them will respond, but then some people will actually have frequent relapsing and steroid-dependent cause. So we do not have predictors of pattern of response to corticosteroid. Therefore, we give therapy to children with significant side effects without actually being able to say that this is the person that will benefit, this is how this person is going to behave when we give the therapy. So, the consequences of that, of course, like I said, is exposure to medicine with major toxic side effects. So we feel the approach to this, because this is a common glomerular disease among children at least, and we know that its inheritance is actually complex. Therefore, the tools to study the disease should actually be population genetics approach. So that's gene and environment interaction. And the best way to study that is actually genome-wide association studies. I will talk about that towards the end of this talk. How about steroid-resistant nephrotic syndrome? Its pathogenesis is unclear. There is no satisfactory therapy. We know that majority of children who are steroid resistant will progress to end-stage kidney disease requiring dialysis or kidney transplantation within 5 to 10 years of making diagnosis, okay? And end-stage kidney disease in most of them. And the sad thing about SRNS is that the major cause is actually focal segmental glomerulosclerosis. And we know that post-transplant, patients with focal segmental glomerulosclerosis can actually have recurrence of the disease, and is actually a major cause of kidney transplant loss. So what we have discovered in the last 20 years is that there's actually this small percentage, depending on the population that you are studying, between 5 to 30%, that are actually due to single gene defects. So when you have something that is due to single gene defect, once you identify that defect, then the thing to do is actually to study the mechanism by which that defect causes disease, and then take it on to developing therapeutics. Of course, there is a large percentage of SRNS and that are actually like SSNS too. So the approach to those ones too will still be gene and environment approach. Okay. So why do I study nephrotic syndrome? And what I typically said is that the reason is that the pathogenesis has not been clearly delineated. And like I said, therapy response is variable. So this is the histology of a kid with steroid-sensitive nephrotic syndrome. Typically, what you see is actually what we describe as minimal change disease. So if you look at this, this is a perfect glomerular tuft, okay? So, but then if you look at the steroid-resistant nephrotic syndrome, the most common thing that you see under the microscope is actually what we call focal segmental glomerulosclerosis. So if you look at this pink area, all these are actually scar tissue, okay? However, if you look at both of them under the electron microscopy, they actually have very similar picture. You know, they have fusion and effacement of the podocyte foot process. Okay, so we do not understand how this lesion evolved. So that's number one. Now, I've talked about corticosteroid and other immunosuppressive that we use. Most of these therapy are non-specific and they have multiple side effects. In addition, some people do respond, some people do not respond. We do not have markers to identify who is going to be a responder and who is not going to be a responder. So unmet needs in nephrotic syndrome, as far as I'm concerned, include one, robust and specific diagnostic tools, and the fact that we do not have specific and non-toxic therapeutic agents. So this is the approach that we use. I know that this is bread and butter for most of the people in this audience. So we use forward and reverse genetics. So forward genetics means we know the phenotype, which is nephrotic syndrome. And then we carry out a number of studies. When Jackie and I were training, the steps were linkage, positional cloning, then identification of the gene. But these days, we actually have a much more efficient workflow thanks to the genomic revolution. Okay, so you carry out that, and then you discover the gene that is responsible for the phenotype. On the other hand, reverse genetics means you actually know the gene, you manipulate the gene, and then you produce the phenotype, which what I'm trying to depict here is an obese mice. I don't think this mice is actually really that big. And then you carry out studies in these mice and figure out the mechanism by which these mice actually develop obesity. Okay. All right, so that is the approach that we use, and that approach has actually been revolutionized with the completion of the Human Genome Project. I would describe Human Genome Project as one of the largest collaborative projects ever undertaken in biomedical research. And the rationale for the great people that designed this study is to have the ability to take global view of genomes, and that this could actually increase and accelerate biomedical research. So the project took about 10 years, as we all know, and less than 20 years since the original publication. Because of advances, we can now accomplish the same task in less than 1 week. That is sequencing a whole genome in less than 1 week and actually be able to interpret and interrogate it. So what is the impact of this on nephrology? we know that has actually given us the ability to unravel single gene causes of kidney diseases. There are over 350 kidney disease genes now that are well known. In addition, there are greater than 100 genome-wide association studies that have been published in the literature. More specifically for nephrotic syndrome, being able to carry all this genomic study, I've now identified over 80 nephrotic syndrome and FSGS genes. And what we have learned from all this finding is that the cellular lineage in the kidney that is actually driving disease pathogenesis is the podocyte. And this is actually a kind of cartoon of the podocyte with all the genes that can cause nephrotic syndrome. And with all these discoveries, there have been identification of some new therapeutic agents. So I will summarize that the impact of the genomic revolution in nephrology generally is actually better classification of disease and understanding of disease mechanism. However, going by what is going on in cardiology, neurology, and other specialty, I think we're actually lagging behind in terms of identifying new diagnostic and therapeutic tools. And the reason for this, I will discuss later on in the talk. Okay, so if you are looking for panel to study genetic kidney disease, this is the kind of list that actually come up for you. So you look through this and actually identify what you think the phenotype is, and basically send this out. This would not have been possible like 20 years ago. So this really has had a big impact on our field. Okay, so let me just describe briefly a recent contribution from our group. So, I talked about the way we approach mapping human disease kidney gene, that you identify the family, you put together your pedigree, you do your phenotype screening, you do your deep phenotype screening, and then you carry out linkage analysis, and then you start direct sequencing of the gene within that region. It turned out that what I've just described can actually take about 2 to 3 years for 2 postdocs working on one project. Now, with the advent of next generation sequencing, the workflow has actually changed considerably. And this is what we actually do in our group now. So, of course, the bottom line is that you still have to be a good clinician. You have to go to the clinic, identify the patient, put your pedigree together, and convince yourself that this is likely to be a familial disease. Then instead of doing all those linkage analysis and all that, you do whole exome sequencing and whole genome sequencing on affected and unaffected individual. You use your bioinformatic pipeline to sort the data. At the end of the day, most of the time, you will end up with 0 to 10 variants, okay? That's when you now bring in your supporting evidence to say which of these variant is likely to be pathogenic. So you can do a modified linkage analysis. You can look for variants in other cohorts, and we are very, very lucky that there are national initiatives that are actually developing cohorts. So example is Neptune, example is Kidney Precision Medicine Project, example is CureGN project. And then you can do your in vitro phenotyping, look at expression in human kidney tissue, and carry out your mechanistic study. So, but here's the question: why should we continue to identify new causes of monogenic nephrotic syndrome? Reasons are: one, we know the cellular lineage driving the disease pathogenesis, so it should be easy to interrogate. Okay, like I said, we still do not have enough specific therapeutic agents in nephrology field, and despite all these advances that I've actually described, actually in more than 80% of patients with familial or hereditary nephrotic syndrome, we still do not have a cause. Okay, so that was what we did, and this is Brandon Lane, as research assistant professor in our group, who actually led this study. Okay, so our a priori hypothesis is that the cause of familial FSGS is unknown in 80% of families, and we carried out whole genome sequencing in 320 individuals from 201 families. Now, before you start thinking that, oh my God, Rasheed is a millionaire, I'm not. What we did was actually to foster a collaboration with a biotechnology company to do this sequencing. And pretty much we did it in very, very ethical way, and they were able to give us a sequence worth close to $500,000 for free. So one of the things that we need to train our young investigators and colleagues to do is to collaborate with biotechnology firm in very ethical way. So we identified a large family with multiple individuals with FSGS and chronic kidney disease. We interrogate their whole genome sequencing. And like I said, our a priori hypothesis is that the disease-causing variant will be rare and it will be biologically possible. So we identified a segregating variant in a gene called RCAN1, and that's regulator of calcineurin type 1. So Dr. Ellis, thank you so much for your pioneering work in using tacrolimus and cyclosporine to treat patients post-transplant. So as you know, there are actually variability in the response to this therapy or the therapeutic levels that you need to reach before you achieve immunosuppression. We use tacrolimus and cyclosporine to treat children with nephrotic syndrome too. Some of them will respond, some of them will not. So we do not know the reason for this. So the panel A is actually showing the pedigree and our sequencing data, and panel B, C, D, E is showing the renal biopsy findings in these individuals. So there are 3 genes in the RCAN family. So there is RCAN1, RCAN2, and RCAN3. So we screened about 1,000 patients with steroid-resistant nephrotic syndrome for variant in RCAN1. And this is through collaboration with other investigators around the world. And we identify 5 additional pathogenic variants in these 3 genes. And all the variants actually disrupt the conserved protein domain of RCAN1, and that's actually what is shown in this figure. So the functional domain are actually the domain that are shown in green and yellow. So as we all know, calcineurin signaling is actually ubiquitous in the kidney, and it's important for maintaining the structure and function of key cellular lineage in the kidney. So we posit that in addition to the fact that variants in RCAN1 can cause single gene disease, it may actually also increase the risk for different chronic kidney disease. So what we did was to examine the association between functional rare variants in RCAN1, truncating mutation, missense mutation in patients with chronic kidney disease and patients with neurological disorders in the Genomics of England database. As you all know, podocytes and neurons are actually very, very similar. So we choose neurological disorders because that's actually the best phenotype that we could get from Genomics England database at that time. And what we found, despite the closeness of the neuron and podocytes, what we found was that rare variant in RCAN1 is actually enriched in patients with chronic kidney disease compared with patients with neurological disorder. So we concluded that RCAN1 to 3 is a single gene cause of nephrotic syndrome and FSGS, but it may also be susceptibility genes for different chronic kidney diseases. And we're actually pursuing that line of investigation currently. Okay, so how does RCAN1 likely to predispose to kidney disease? So RCAN1 regulates calcineurin activity by forming a complex with its catalytic domain. So we examine the effect of RCAN1 gene deficiency in podocytes on calcineurin activity and cell survival. So panel A is showing you the knockdown that we're able to achieve, and the same thing is actually quantified in figure B. Okay, so what we did next was actually to look at calcineurin activity when we knock down RCAN1. And what you can see is that in the knockdown cell line, we actually have increased calcineurin activity compared with the control podocytes, and that actually translates to increased apoptosis in the knockdown cell line compared with the wild type. So we concluded that loss of RCAN1 increases calcineurin activity and reduces viability in human podocytes, and we also showed that the increased apoptosis can actually be rescued by calcineurin inhibitor. So we did the same experiment with the mutation that we found in the family with FSGS, and pretty much we actually recapitulate the same thing. And that's what all these figures are showing. So just look at the increased calcineurin activity in our mutant and increased NFAT luciferase activity, showing that there is uncontrolled regulation of calcineurin activity, and that actually translates to increased apoptosis, and we were able to rescue that by treating the cells with calcineurin inhibitor. So, so this is just the cartoon showing the wild type ARCAN1. As you can see, you do not have many signals of apoptosis here compared with the missense mutant. You can see the green and the red, which are markers of early and late apoptosis. And then when we treated with calcineurin inhibitor, we actually rescued that phenotype. So it turned out that we did 3D modeling of the mutation that we found, and we showed that alteration to the positioning of serine 163 residue is a target of GS3K kinase. So we treated with, we inhibited GSK3B kinase, and we were actually, just like we did with the calcineurin inhibitor, we were actually able to rescue the phenotype. Okay, so the summary of this part of the talk is that mutation in ARCAN1 can cause FSGS. Loss of function mutation lead to uncontrolled calcineurin activity and reduced podocyte viability, and the aberrant podocyte phenotype can be rescued by calcineurin inhibitor. In addition, we showed that this mutation actually disrupt GSK3 kinase, and we used GSK3 kinase inhibitors to rescue the phenotype. So targeting ARCAN1 regulatory molecules like GSK3B and other kinases may represent novel therapy for FSGS and chronic kidney disease. We know that the signaling from GSK3B is ubiquitous in the body, so may not be a viable target, but we are looking at molecules upstream and downstream of GSK3B in order to identify a novel therapeutic target for FSGS. Okay, so the next part of the study, I will now dive into complex inheritance. And I think the best model for complex inheritance to discuss in our field today is actually to look at the story of APOL1. So this is the cartoon that I used to differentiate between Mendelian disease and complex inheritance. So if you look at the left, Mendelian disease, they are supposed to be very, very rare, and they are very hard to identify genetically. On the other hand, complex disease, they are fairly common, and the only thing is that, the common variants on their own actually do not cause disease. They predispose to disease in the presence of additional genetic factors and environmental factor. So a very good example of this is actually APOL1 and chronic kidney disease in people of African ancestry. So we know from epidemiologic study that people of African ancestry have about 4 times higher age-adjusted risk of end-stage kidney disease. And FSGS is the most common of those diseases. So what Dr. Kopp and other investigators did in 2008 is actually to use a modified GWAS that they call mapping by admixture linkage disequilibrium to identify the genetic factors that may be driving this disparity. Okay, so long story short, they identified a region in chromosome 22, which Dr. Martin Pollack from Harvard actually fine mapped later and identified that the variant that is actually driving this excess disparity are actually 2 variants in APOL1 that they called G1 and G2. Okay. So they showed that homozygous or compound heterozygous variants in APOL1 called G1, 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 G2, or G2, G2 are major driver of excess of CKD in African Americans, especially FSGS. And they showed that G1 and G2 likely rose to high population prevalence because of the protection against deadly African sleeping sickness thousands of years ago. So it turned out that the same variant that is protecting people from developing sleeping sickness later on actually predisposed to chronic kidney disease. And the penetrance of this variant is so high to the extent that people actually feel that we should have a new disease category that we call APOL1 kidney diseases. So this is the odds ratio of having high-risk APOL1 genotype and kidney disease. So primary FSGS, 17 times odds ratio. HIVAN, another viral-associated disease, 29 times, and sickle cell nephropathy, about 3 to 6 times. So this is really a large effect size for a risk factor for a kidney disease. So people got excited about it. So the next question is, how does APOL1 predispose to kidney disease? So APOL1 is an immune effector that protects against sleeping sickness in human and non-human primates. And the G1, G2 variant cause trypanolysis and therefore protect against African sleeping sickness. And basically, in people with the G1, G2 genotype, the genotype actually for the protein, the defective protein actually form pores in mammalian cells, affecting cation flux and cytotoxicity. This cartoon is actually showing some of the disease mechanism that people have actually elucidated over the last 10 years. So cytokine-mediated increased expression of defective protein, cation flux, activation of inflammasomes, among others. Okay. So when you identify such things, you look at it further. So it turned out that APOL1 gene in itself may actually be functionally redundant, because there are people who do not have copies of APOL1 and they lead perfectly normal life. So this is actually a report from my very good friend, Dr. Duncan Johnston, when I think he did this and when he was actually at so me and Duncan trained together in Michigan, and then he moved to UPenn. So they identified this family in India who actually do not have any copies of APOL1 at all. And members of this family actually lead completely normal life. They are members of the family who were 80 years to 90 years at the time they carried out the study. And there was actually a similar report in England Journal of Medicine by another group. So that actually makes it easy, because if you have a gene that is relatively functionally redundant, that means that you can actually target the gene, knock off the gene itself completely, or basically modulate the function of that gene. And that's exactly what people are doing. So in less than 10 to 14 years, there are actually 3 agents that are in clinical trial for APOL1 associated kidney disease. So one of it is actually a compound that is targeting cation flux across membrane. There is another study that they've completed phase 1 of the study, and they're going to start phase 2 soon. That is actually using antisense oligonucleotide to silence APOL1. And then there's another study that is actually targeting expression of APOL1 and its downstream effect on cytokines. That's the Janus kinase start inhibition trial, justice trial, that is actually being led by Dr. Opeyemi Olabisi here at Duke. Okay, so those are some of the agents, some of the approach. But for this talk, I will actually concentrate on the cation flux across membrane, because there's actually a compound now called inaxaplin that is currently in phase 3 trial. So the phase 2A clinical trial, what they did in this study is actually to show that this inaxaplin inhibits APOL1-mediated pore formation and aberrant channel-mediated ion flux in cells carrying the G1, G2 genotype of APOL1. So they did a thorough study, first did it in vitro, did it in a humanized mice that is actually expressing APOL1, because naturally, mice actually do not have APOL1 gene. And then they carry out human study. It's actually a single group open label phase 2 clinical study of this compound in 13 patients with 2 APOL1 variants, FSGS and proteinuria. Okay, I think I will skip this slide. And that the result of that study was actually very, very remarkable. So they gave these individuals the medication and actually followed them up for 13 weeks. And as you can see on the left panel, 12 out of these 13 individuals actually have substantial reduction in their proteinuria and proteinuria is a marker of disease activity in glomerular disease generally. And then there's this single individual that did not respond. The reason for that, we do not know, but I think there are actually possible explanation for that. And then they followed this individual to week 13, and the reduction in proteinuria was actually sustained. So the bottom line is that in less than 10 years after discovery of APOL1 CKD variants G1, G2, we now have at least 3 agents in clinical trial, and this demonstrates clearly the power of genomics in drug discoveries, considering the fact that from the pharmaceutical company, when they are trying to find new compounds, from the time that they start to when you see the drug in clinical trial, you are probably looking at about 25 years. This happened in less than 10 years. And then moving forward, there may actually be other possible indications for inaxaplin in the treatment of glomerular disease. So I did a news and view for Nature Nephrology for that. And that's actually what is shown in the box on the right side. Okay, now still on APOL1 story. So we know that 13% of people of African Americans living in this country carry the high-risk genotype. In fact, it is worse when you go to West African population. So I'm originally from Nigeria, and my tribe in Nigeria is called the Yoruba tribe. About 30% of Yorubas in southwestern Nigeria actually carry the high-risk APOL1 genotype. In fact, if you move to the southeastern part of the country, it's actually close to 50%. However, we know that the lifetime risk for developing CKD in people with high-risk genotype is probably about 15%, right? So telling us that there may be, disease modifiers. And I think some of these modifiers have been shown clearly. So we know that people with high-risk APOL1 genotype, when they develop HIV or COVID-19, the people with high-risk genotype actually develop, the odds of developing FSGS is actually very high. Now, the problem with this is that the problem with genetic modifier study is that it's actually difficult to identify a large cohort. So people have published all sorts of studies on genetic modifier, and most of them are actually false positive because they do not have the power to detect the effect of the modifier. Okay, why do we care about modifiers at all? So we know that if we know modifiers, it can actually lead to proper risk stratification in transplant selection of living-related donor, and then it can actually predict allograft longevity too. There's actually a study that is looking at, a national study that is looking at that now called the Apollo study, okay? And so this is what my very good friend, Simone Sanakaki from Columbia University did. He basically assembled many investigators in this field, built a collaborative group, including us, and actually look for modifiers for APOL1 within APOL1 gene. Okay, so it turned out that there's actually another variant in APOL1 gene called the N264K gene, and people looked at it and found that if someone actually co-inherits this variant with the G1 or G2, at least that's what they showed on in vitro, if you co-inherit it with the N264K, it can actually abrogate the effect, the cytotoxic effect of the G1 and the G2 allele. And that's actually what is shown here. So look at the G1, G2, so that's cytotoxicity. This is GO with N264K. So no cytotoxicity is actually very low. But when you look at someone who actually co-inherits G2 with N264K, the cytotoxicity level is actually very similar to the wild type, suggesting that this variant can actually abrogate the unwanted effect of G2. This is actually a very, very remarkable and powerful genetics. Okay, so, and this is another case report. I won't go with that. So what Dr. Sanakaki did was actually to identify 528 patients with high-risk APOL1 genotype, and then 2,606 individuals with high-risk APOL1 genotype with no disease. And then his hypothesis is that if that variant is truly protective, that variant should actually be more common in individuals with high with high-risk APOL1 genotype without disease compared with patient with disease. And then he confirmed this in another cohort. So the bottom line is that he showed that there is strong protective effect of the A allele in cases versus control. And if you look at this, so you have your G1, G2 control here. This is the frequency of the protective allele that's shown in pink. If you look at the G1, G2 cases, you will see that the variant is actually not common in those one. But if you look at the G2, G2 controls, that's people who actually have the high-risk genotype but no disease. Look at the prevalence. And then the other thing that it showed is that this variant is probably co-inherited with the G2 allele, because in patients with G1, G1 control cases, it's actually absent. So this is really powerful, and he reproduced the same thing in a CKD cohort. And the direction was actually consistent in that group too, and it showed about 3 times, protective effect. So it turned out that another group used another cohort and showed exactly the same thing. Okay, so what it means now is that the traditional way by which we classify APOL1 disease is that we have what we call the low risk, GOGO, and then we have the undetermined risk. Those are people carrying one allele. And then we have the high risk, that's people carrying the 2 allele, either G1, 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 G2, or G2, G2. So based on these findings, we actually need to reclassify, and this is actually making life, much more complex when we get genetic results. So this is a review that I wrote, talking about the implications of this finding. So the summary is that there is strong protective effect of the N264K variant against G2-mediated FSGS and chronic kidney disease. And this variant is probably inherited with G2 and G haplotype. The effect is not seen in the more common G1 allele. And the absence of effects on G1 suggests the existence of additional modifiers yet to be discovered. And Dr. Simone Sanakaki, in collaboration with us and other investigators, are actually looking at this. Okay, so quick problems, prospects, and conclusion. I think we need to do this. So there is changing landscape with advances in genomic technology, exciting progress in nephrology in the last 10 years. We have APOL1 specific compounds. We have gene therapy for oxalosis. We have compounds targeting another cause of FSGS called TRPC6 and others. However, kidney disease is still lagging. The 5-year average for drug approval for kidney disease is less than 1% of all approval, well below the 10 to 15% for oncology and cardiovascular disease. Why is this so? So at least for glomerular disease, I believe that there are 3 major problems. We do not have good cell lines, and we have animal models that do not faithfully recapitulate human phenotype. Murine models are actually not very good model for studying glomerular diseases, in my word. Yes, we carry out different manipulation, knock out, different genes, treat them with different compounds, and then we get the phenotype that we desire. But the point is that that phenotype is not physiologically relevant. So we need to look more closely. And like I said, this is the culprit. So this is what happened when we do gene discovery. Once we discover the gene, we have to do the mechanistic study in animal model. And for glomerular disease, everything just goes downhill from there. Okay, so what are the prospects and solutions to that? We know that people are using, thank God, we can now generate pluripotent stem cell from patients with kidney disease. And then we can use the iPSC for compound library and toxicology screening. And also, there are some people that are actually working on kidney on a chip platform. That's actually what we did for the RCAN1 variant that we found. We got cells from the family, generated iPSC, and showed that this iPSC actually has characteristics of podocytes because they express synaptopodin robustly. And the same changes that we found in our artificial in vitro data that I showed earlier on, we actually showed exactly the same thing in the iPSC podocytes. Okay, so moving forward with the study, we are going to leverage our findings so far to predict response to calcineurin inhibitors in patients with nephrotic syndrome. Can we explain why some people can we use this to actually explain why some people respond to calcineurin inhibitor and others do not? Okay, and then we are going to use the iPSC for pathway analysis and compound discovery. Okay, so we are doing this in collaboration with many groups. We have collaborators from University of Michigan and University of Miami, and then my colleagues at Duke. Okay, so to conclude, advances in genomics and bioinformatics pipeline is speeding up discoveries of genetic causes and genetic risk factors for glomerular diseases and other CKDs. We have over 350 single gene causes of chronic kidney disease. We identify ARCAN1 mutation as a new cause of autosomal dominant FSGS. Pathogenic variants in ARCAN1 can cause reduced cell viability and uncontrolled calcineurin activity. There are promising drug targets from GWAS in clinical trial. More studies are needed to have precise characterization of those who may benefit from the new and specific therapeutic agents. iPSC-derived cells and kidney organoids are potent tools for future mechanistic studies and compound discoveries. And with that, I will need to acknowledge all the collaborators that have been working with us over the years. In particular, please let me acknowledge Dr. Michelle Nguyen, my late mentor. She was a great mentor and a great sponsor. She left too soon, but we actually continued the great work that she started. She was the one that discovered mutation in TRPC6 as a cause of autosomal dominant FSGS, and there are actually compounds targeting TRPC6 that is in clinical trial now. With that, I will stop and I will be happy to take questions. Thank you very much.